My dear students, today I will be giving you a lecture on cytoskeletons and cell membrane. First, let us take off cytoskeleton. These are extremely minute, fibrous and tubular structures found within the cytoplasm, which form the structural framework inside the cell. These structures maintain the shape of the cell and its extension, and also help regulate orientation and distribution of cell organelles, intracellular transport, and movement of cells. The existence of these fibrous structures in the protoplasm of the cell was postulated in 1928 by Kolzov. He observed that each cell was a system of liquid components and rigid skeletons. The skeletal fibers generated the shape of the cell, though they were difficult to see as they were very thin. The observation under the electron microscope revealed the presence of cytoskeletal fabric in most eukaryotic cells. They are formed by microtubules, microfilaments, and intermediate filaments. First, let us take off microtubules. The microtubules are structures universally present in the cytoplasm of eukaryotic cells and are characterized by their tubular appearances. De Robertis and Farnsey observed these tubular structures for the first time in myelinated fiber in 1953. These microtubules are straight and uniform in size with about 25 micrometer in outer diameter and several micrometers in length. Each microtubule is surrounded by low electron density region where ribosomes or other particles are absent. Microtubules have wall consisting of individual, linear or spiraling filamentous structures about 5 micrometer in diameter. These structures are made of about 13 subunits with a center-to-center spacing of 4.5 micrometer in cross-section. The principal protein of microtubule is the tubulin and is a dimer of 1,10,000 to 1,20,000 Daltons. It consists of two different monomers, tubulin A and tubulin B, which have similar molecular weight. The assembly of microtubules from the tubulin dimer is a specially oriented and programmed process. There are sites of orientation in the cells from which the polymerization is directed. During the interphase and metaphase stages, the quantity of polymerized tubulin, cytoplasmic microtubules and spindle microtubules is high but low at prophase and anaphase stage of cell division cycle. The polymerization of tubulin monomers in a cell is achieved by the action of cyclic AMP-dependent kinase. The microtubules are in equilibrium with the free tubulin in cell. Next, let us take up cilia and flagella. The cilia and flagella are derived from special assemblies of microtubules. They are fine hair-like protoplasmic processes of the cells which are capable of producing a current in the fluid medium for locomotion and passage of substances. Flagella are longer, about 100 to 200 micrometer, and fewer in number, that is from 1 to 4 in number, as compared to cilia, which are smaller, 5 to 20 micrometer, and numerous. The flagella are locomotory organs of many protists motile algae, spermatozoa of animals, bryophytes, and pteridophytes. The cilia and flagella are structurally similar and possess similar parts, that is, the basal body, rootlets, basal plate, and sap. The basal body or kinetosome is also called basal granules or blepharoplasts and is found embedded in the outer part of the cytoplasm below the plasma membrane. It has structure consisting of 9 triplet fibrils present on the periphery without a central fibril. The rootlets are striated fibrillar outgrowths which develop from the outer lower part of the basal body. They are meant for providing support to basal body and are made of bundles of microfilaments. The basal plate is an area of high density which lies above the basal body at the level of plasma membrane. The central fibrils are developed from this area. The shaft is the hair-like projection of the flagellum or cilium. The shaft is covered on the outside by sheath, which is the extension of plasma membrane. Internally, it contains a semi-fluid matrix having an axoneme of 9 peripheral doublet fibrils and 2 central singlet fibrils. This type of arrangement is called 9+2 or 11-stranded. The 2 singlet fibrils present at the center is covered by proteinaceous central sheath. Next, we'll come to microfilaments. They are ultramicroscopic, long, narrow cylindrical rod-shaped like protein structures present in plant and animal cells. They range from 5 to 7 micrometer in width and represent the active or motile part of the cytoskeleton and appear to play the major role in cyclosis and amoeboid motion. The contractile proteins actin and myosin, as well as tropomyosin, troponin C, alpha-actin, and other proteins which are found in muscles are present in microfilaments. These microfilaments are often associated themselves to form hexagonal bundles, but they may also occur in parallel bundles or loose network. They generally lie at the soul-cell interface as well as below plasma membrane. They are found connected with spindle fibers, endoplasmic reticulum, chloroplast, etc. In animal cells, they are found to be associated with clavispirals during mitosis. The microfilaments are responsible for the cytoplasmic streaming or cyclosis, which is found in all the cells. This movement produces the displacement of various organelles in the cell. The microtubules provide the framework for cyclosis, but the actual motive force is provided by the action of microfilaments in the cells. The microfilaments play an important role in chains of cell formed during development and differentiation. It also aids in the movement of fibroblasts through undulations of plasma membrane. It is also responsible for changes in plasma membrane during endocytosis and exocytosis. Pigment granules, chloroplasts, and other cell organelles are able to change their position inside the cytosol due to the activities of microfilaments. Next, we have intermediate filaments. They are solid unbranched filaments of about 10 nanometer in thickness, which are formed by a variety of proteins and often form a network. They are grouped into 4 main types: keratin filaments, which are present in epithelial cells and contain the alpha keratins. They are the most complex class of intermediate filaments, which are found to anchor to the cell surface and tend to converge upon the desmosomes. neurofilaments contains 3 polypeptides ranging between 2 lakhs and 6,80,000 Daltons and are sensitive to proteolysis. They, along with microtubules, are the main structural elements of axons and dendrites. Next, glial filaments, which are found throughout the cytoplasm of astrocytes and are composed of proteins of 51,000 Daltons with very acidic properties. These intermediate filaments are found only in astrocytes but not in the oligodendrocytes of the brain. Heterogeneous filaments have similar morphology and localization but contain variety of proteins. They contain desmin filaments, which in muscle constitute a cytoskeleton that holds the myofibrils within the muscle cells. Next, we will come to cell membranes. Cell membranes or biological membranes are organized assemblies consisting mainly of proteins and lipids. The structure of all the biological membranes have many common features, but small differences exist to accommodate the varied functions of different membranes. Phospholipids are the major class of lipids that form the membrane. This lipid molecule has a central, somewhat polar carbohydrate moiety linked to highly polar groove, we call it head, and to two nonpolar long hydrocarbon chains, we call it tails. The essential feature is a polar head groove to which hydrocarbon tails are attached. Cell membranes contain many protein molecules, about 50% by weight, and the membranes having different functions contain different proteins. The fundamental role of a membrane is to separate the contents of a cell from the environment. The enclosing membrane of a cell should also be permeable so that the cell must be able to take nutrition from the surroundings. But the permeability of the membrane must be selective so that the concentration of compounds within a cell can be controlled and intracellular concentration and extracellular concentration remain different. We have different models of cell membrane. First, let's take up Gautier and Grendel's bimolecular lipid leaflet model. Gautier and Grendel in 1925 proposed that the cell membrane was formed by a bimolecular lipid sheet. The polar ends of the lipid molecules of one layer were directed outward from the cell towards surrounding plasma, while the polar ends of the lipid molecules forming the other layers were directed inward towards the cell hemoglobin. The validity of the model depends on the assumption that all the erythrocyte lipids are in the plasma membrane. All these lipids were extracted using their acetone procedure, and the average surface area of the cell was accurately estimated. Next, we have Danieli and Dapson membrane model. Danieli and Dapson suggested that the plasma membrane might be composed of two lipid protein bilayers, one facing the interior and the other facing the exterior of the cell. The electrostatic interaction between the polar ends of each lipid molecule and charged amino acid side chains of the polypeptide layer maintain the association of surface proteins and bimolecular lipid layers. Two distinct layers of proteins are present in the model. They proposed that plasma membrane would exhibit selective permeability as they would be capable of distinguishing between molecules of different sizes and solubility properties, and also between ions of different charge. By the early 1950s, several modifications were made in the Danieli-Dapson membrane model. There was suggestion that glycoproteins might be adsorbed to the outer membrane surface, thereby accounting for the antigenic properties of cell membrane. Next, we have Robertson's unit membrane. According to Robertson, the unit membrane is made up of bimolecular lipid leaflet, which was sandwiched between the outer and inner layer of proteins organized in the pleated sheet configuration. This arrangement was presumed to be basically the same in all the cell membranes, though he acknowledged the specific chemical differences between the membranes. He proposed that the pattern of molecular organization was fundamentally the same. Robertson extended his unit membrane model to include the notion that continuity exists between the membranes of the nuclear envelope and the plasma membrane via the endoplasmic reticulum. Next, we have the fluid mosaic model of membrane structure. The most widely accepted model of cell membrane is the fluid mosaic model developed by S.J. Singer and G. Nicholson in 1972. They proposed that a membrane was flexible bimolecular lipid layer which was interrupted by proteins. The proteins present in the cell membrane can be grouped as the peripheral or extrinsic proteins or integral or intrinsic proteins. The peripheral proteins are loosely attached at the periphery of the membrane through the polar surface of the lipid. They can be more readily removed and are rich in amino acids with hydrophilic side chains. These side chains allow them to interact with the surrounding water and polar surface of lipid bilayer. The peripheral proteins located at the cell exterior membrane surface often contain chains of sugar glycoproteins. The integral proteins contain both the hydrophilic and hydrophobic regions and are found to be penetrating the interior of the membrane or span the membrane entirely. The hydrophilic portion of the protein interact with the polar heads of the lipid molecules at each surface of bimolecular leaflet. Portions of the protein that are buried in the hydrophobic part of lipid bilayer are rich in amino acids with hydrophobic side chains. These side chains are believed to form hydrophobic bonds with the hydrophobic tails of the membrane phospholipid. The integral proteins which span the entire membrane contain the two outer regions that are hydrophilic and a central region which is hydrophobic. There is a differential distribution of proteins in various regions of plasma membrane within a single cell. This irregular distribution of membrane protein is known as membrane asymmetry. Some proteins of the membrane are shown to possess enzymatic activity. The enzymes located on the cell membrane may be ectoenzymes or endoenzymes depending on the phase of membrane containing the enzymes. Ectoenzymes are those enzymes whose catalytic activity is associated with the exterior surface of plasma membrane, while the enzymatic activity of endoenzyme is associated with the interior of the cell. Most of the ectoenzymes of cell membrane are glycoproteins. Finally, we have the conclusion. The cytoskeletons, which are made up of filamentous proteins, help cells in maintaining a safe and internal organization. The three major classes of filaments, microtubules, microfilaments, and intermediate filaments, constituting the cytoskeleton system of the cell, also provide mechanical support that enable cells to carry several essential functions. The cell is delimited at its surface by plasma membrane, which actively participates in the exchange of material between the cytosol and surrounding environment. The movement of materials across the cell membranes may be achieved by active or passive mechanism. It also plays a role in maintaining the physical integrity of the cell by mechanically enclosing the inner contents of the cells. The eukaryotic cell also possesses internal membranes which enclose different organelles present in the cytoplasm and control the exchange of essential cell components.